Right. So hello everyone. My name is Giacomo Marino, and I'm a bioinformatics software engineer in the Mayan Lab, the Icon School of Medicine at Mount Sinai. I'm a member of the DRC team in the Links DCC. And today I'll be speaking about one of our new resources called RummaGEO. So just to begin with an overview of the project, the main goal of this project was to automatically compute signatures from uniformly aligned bulk RNA-seq studies extracted by GEO from our resource ArchIS4. And we do this through identifying conditions using metadata string embeddings and then k-means clustering. We can also sort of verify our samples partitioning at the data level using a silhouette score. And additionally, we can extract functional terms from the GEO sample and study metadata, as well as associated abstracts and summaries. And this we can use to help summarize gene set search results. To serve all of these gene sets, because it's quite a large collection, we utilize and adapted the framework that was built for RummaGene, which has automatically extracted signatures from PubMed Central articles, which was developed by Daniel Clark. And this allows us to serve those gene sets for enrichment analysis, PubMed search, as well as metadata search. So this is an overview of the workflow of RummaGEO. So we start with all of these aligned, uniformly aligned human and mouse studies. And then we have some constraints on the number of samples, as well as filtering out samples that are likely from single cell experiments. And then, like I said, before we use these sample metadata string embeddings of some of the key sample attributes, as well as k-means clustering to partition those samples into various conditions. And we can create titles that describe those conditions by looking at shared words across those group samples. After this, we can look at, and see if we find any keywords that would denote a normal condition, such as wild type, control, and then use Lima-Boom to actually compute differential gene expression signatures and extract up and down gene sets based on the adjusted p-values. We'll basically just discard sets where there weren't enough significantly differentially expressed genes, so less than 5. And then for those that are greater than 2,000, actually incrementally lower the threshold to get below that number. So this is an example of this metadata attribute embedding that I was speaking of. So on the top row here, we have the metadata string embeddings as a PCA for this study that was looking at gallbladder, tumor, and normal tissue from 4 different patients. You can see in the top, we achieve a pretty nice separation of the tumor and normal. And what's differentiating them here really is just the patient number that we see in this metadata. Whereas in the data level, we see for 2 of the patients, it looks like the tumor and normal are grouping together quite well. So this may speak to the data's quality. But this is part of the reason why we chose to sort of use the metadata to actually partition and group the samples. And then we can actually evaluate how well that aligns with the data afterwards, which is done with this data level confidence score with this silhouette coefficient. And this basically reflects the tightness and separation of the clusters with a value of 1 to -1. And we can see here on the right, this is the distribution of silhouette coefficients across human and mouse. And we see this sort of bimodal distribution where sort of a lot of the studies cluster quite well around that 0.5 or above, and then some cluster not quite as well or don't have as much agreement at the data level around 0. So looking at sort of the contents of the RummaGEO database, we can sort of observe a few different things. One being that this first row or column here, we have human on the top and mouse on the bottom. This is looking at the genes and then how many gene sets they are contained within. So we see a majority of genes are contained in very few gene sets, and a few are contained in many gene sets, like up to around 10,000. We see in the figure there. And then we have a distribution of the gene set lengths, with the y-axis is log scaled. So there's sort of a lot of smaller gene sets and then sort of an equal distribution moving outward towards 2,000. And then another sort of thing we can look at is how many gene sets are we getting per study. And we can see this periodic nature of this chart, and that's sort of arises from the fact that a certain amount of conditions, if you do some combinations, can lead to a certain amount of gene sets. So, for instance, 3 choose 2 or 4 choose 2, and that's what's leading to these sort of peaks. We can also look at the groups per study, and here we see sort of something interesting in the fact that there is sort of a bias towards an even number of groups. You can see 3 and 4 being equal and 5 and 6, but the majority being sort of two experimental groups there that are being compared. And we can also look at the number of studies per year, and this was taken from sort of a mid-year 2023 release of ArchES 4, and we're currently updating the resource. So you could see there's some drop off there. Um, and then looking at the actual metadata from these studies included in the RummaGEO database, we can identify a variety of functional terms, including tissues and cell types, cell lines, diseases, and genes. And we can look at both how many sort of unique occurrences we have of those, as well as the total amount contained within studies. So here we can see really the most common functional term that we're pulling out are definitely the tissue and cell types, but many other functional terms can also be extracted. And from those genes, we can also look for kinases and transcription factors, which we'll use for benchmarking later on. And we can see that we have quite a sort of comprehensive view. Those transcription factors, about 550 unique across for both human and mouse, and about 200 kinases for human and mouse as well. Visualizing all of these gene sets for both human and mouse, we can see that there is sort of a fair amount of separation for human and mouse with some sort of density of where they overlap here at the top. So this is looking at sort of harmonized gene symbols between both human and mouse here. And then we can also color this UMAP by various metadata attributes that we previously identified. So on the left here is by the 10 most common tissues. And we can sort of visually observe that we see some coherency in clusters of these tissues here that we see from the signatures. And then on the right, we have the 10 most common diseases that were identified. And again, maybe it's slightly less coherent, but we definitely see some clusters of these various diseases that we identified in the GEO metadata. Another thing that we can do is look at where RummaGEO gene sets fall in the enricher gene set space. So on the left is just the enricher gene set space. And then on the right, we overlay all of the RummaGEO gene sets on top of that. And we can see that most of this overlap is in this area here, which is actually corresponding to the enricher crowd category, which has a lot of user submitted GEO signatures. So this makes a lot of sense. And additionally, we can color this by transcription factors and kinases that we saw before. So this is a little bit more sparse, but we can sort of observe some clusters of these transcription factors and various kinases here on the UMAP as well. So with those transcription factor gene sets, we know that they're sort of in there. They're in the signatures. So one approach that we tried was to create a correlation matrix of coding genes from the randomly selected RummaGEO signatures. And then we take the top 200 correlated genes with each transcription factor. And this allows us to create a transcription factor gene set library from RummaGEO. And we can benchmark this with a variety of libraries. And we see that the recovery is actually quite good for transcription factors in that it's ranking the correct transcription factor very highly across many of these benchmarking libraries. And we can use a similar procedure to create a kinase gene set library from RummaGEO. And though this doesn't perform quite as well as the transcription factors, we can still see better than random recovery of those kinases. Another sort of interesting analysis that we conducted was to take the human, in this case, RummaGEO gene set library and transpose it. So now we're looking at genes instead of gene sets. And if we cluster this collection of genes and what gene sets they're in, then we find these, in the case of humans, 77 functional groups. And we can then look at the functions of these clusters from a select set of enricher libraries and see if we're seeing consistent, significant enrichments. And we can see for a majority, about over half in the humans case, we have these sort of quite clear functions for what those clusters correspond to. And then some of the unlabeled clusters, we can see sort of correspond, many of those genes are from the same chromosome, or perhaps they have sort of multiple functions, so it's not quite as clear. But this is a sort of an interesting analysis. And by looking at these clusters, we can also perhaps illuminate some knowledge about understudied genes as well. And so this is the same analysis done for the mouse collection of genes. And in this case, there were 67 functional groups. And for those, about 2/3 had pretty clear and consistent enrichments for a certain function. So just moving on to the actual site now, it currently contains 135,000 and 158,000 human and mouse gene sets, respectively, from over 23,000 GEO studies. And we have sort of three main methods that you can actually query this data, which are the gene set search, PubMed search, and a metadata search. And this is all currently available at RummaGEO.com. So feel free to explore the site afterwards. After you submit a gene set, we'll automatically detect the organism. So that's either human or mouse, depending on the gene symbol. So there's separate backgrounds for those. And then you'll be served with these matching signatures with the enrichment statistics. Clicking on any of the condition titles will show you actually the metadata attributes and the samples corresponding to that condition. And additionally, you can click on the overlap or the gene set size to look at the actual genes. You can copy those to the clipboard or submit them to Enricher or back to RummaGEO as well. Sort of another thing that we did was looked at these functional terms that we could extract from PubMed abstracts associated with these studies or from the GEO summaries themselves. And then we sorted those into categories using an LLM. And these are 4 categories. So we have disease phenotype, biomolecules, tissue/cell type, and pathway or biological process. And here we can actually look at the enrichment of those terms in unique studies that were enriched with your inputted gene set. So you can get a sense of the overall sort of what are those studies entailing that were enriched. And we'll show a bar chart that visualizes the negative log 10 of the p-value. Additionally, below that bar chart, we can see the actual terms and then the counts that lead to that p-value for the Fisher's test, as well as you can click any of those terms, and then you'll be shown a list of all the gene sets that corresponded to those that led to that term enrichment. And below that, we also have just a word cloud to help visualize those terms as well. The last sort of tab on these gene set search page is concerns enricher terms. So we pre-compute significantly enriched terms from selected libraries, including CHIA, KEGG, WikiPathways, MGI, GoBP, GWAS, and the Human Phenotype Ontology. And then we can look where these significant enricher terms rank among the top 500 signatures. And we can compute the significance of that with the KS test. So this helps show what are those actual highly ranked gene sets being enriched for generally. And then speaking sort of to what Avi had alluded to with the integration of LLMs, one thing that we integrated here on RummaGEO was this hypothesis generation module. So here you can actually click this little hypothesis button when you do the gene set search, and you'll be prompted to input a textual description of the gene set you submitted. And then it will take that description as well as the description of the GEO study, and then enrich terms from the overlapping genes from those 2 gene sets to create some hypothesis about why that overlap may exist. You can also filter this table by a search term and download all the results as a TSV. And then for PubMed search, you can search for the PubMed API and then get returned with papers that have corresponding gene sets in RummaGEO. So you can expand that on any of these after you search. And then we also have a metadata search to directly search that GEO metadata, and then we'll sort of bold the search term in the results. So you can grab maybe specific gene sets you're interested or signatures you're interested in. We also have all these gene sets available for download, as well as the co-occurrence matrices that I referred to earlier. There's also a user manual help page where all of the functions of the site are explained. And in the future, we will like to add up and down gene set signature search, since we have these paired signatures. We'd also like to auto update the site as new studies are aligned for ARCHES-4. Certainly, there's a lot we can do to expand this gene-centric analysis that I showed before, and perhaps also serve interactive view maps on the results page, and also add better ways to prioritize or more ways to prioritize and sort the results of a gene set search. And finally, we can also cross RummaGEO and RummaGene to discover unexpected associations between studies. And with that, I'd like to just thank everyone in the Mayan lab, in particular Daniel and Eden, who worked a lot on this project. And also thank you all for listening.